So, good morning, everybody. Today, I will be talking about pituitary and thyroid glands in fishes, the two most important endocrine glands of the endocrine system, which need to be studied extensively. In any organism, uh, this particular system is absolutely critical and immaterial for the survival and of the organism. So, as you know, endocrine system means they don't have any ducts. Am I audible? If somebody could confirm.
Yes, sir.
Yes, sir.
Okay. So, endocrine system, as you know, they are systems which they are particular glands which don't have ducts. So, that is, they directly release their secretions or their hormones into the blood. Also, we know that this endocrine system it starts the most important, the master gland is the pituitary gland, and this pituitary gland is connected to the hypothalamus, and this hypothalamus actually sends a bunch of nerves and releasing factors, releasing hormones to the pituitary, and this pituitary then releases in turn, gets stimulated and release some other hormones in return, and these hormones go to their target organs, and from the target organs it can again induce secretion of another set of hormones. So hormones are the response, and then gradually from the target organs, if the levels of hormones increase, then there is a feedback system which works along with it. So I will start sharing the first slide of the PDF from the book Khanna and Sharma, the fisheries. That is the only and the best book which is currently available for students. So if you look at this particular diagram, this is a hypophysis. Pituitary is also known as hypophysis of the fish anguilla. You can see from in this region, this particular organ here. Follow this arrow. This particular region, this is the posterior pituitary or the neurohypophysis. The connections are with the hypothalamus. So there is a hypothalamus hypophyseal system. The pituitary is also known as hypophysis. So, and it's connected with the hypothalamo hypophyseal system. So you have the neurohypophysis. This is the it has got lots of nerve endings present over here, and this is also known as the posterior pituitary. And you have the adenohypophysis. It has got regions known as pars intermedia, pars distalis. So these are the two regions of the adenohypophysis. And this particular region, the pars distalis, has got lots of different types of cells. As you can see, the proximal regions have got cells known as somatotropes. Proximal region has got somatotropes. And this region, you'll find cells in this region where my arrow is moving. You'll find cells known as corticotropes. Then in this region, the rostral or pars distalis, in the rostral pars distalis, You will have small cells known as thyrotropes and prolactin cells. So the proximal pars distalis has got cells known as gonadotropes, somatotrops, and in the rostral pars distalis, you will find corticotropes, thyrotropes, and prolactin cells. So this is the basic picture of a hypophysis. So as you know, it has got two main parts, that is the neurohypophysis, which you have seen, and the adenohypophysis. This particular region here is the neurohypophysis, all the nerve endings from the hypothalamus, and this whole region is the adenohypophysis. We know that the neurohypophysis is derived from the brain, and the adenohypophysis, it develops as an ectodermal evagination of the roof of the buccal cavity, that is during development. The pituitary is situated ventral to the brain, just behind the optic chiasma. On the bottom side of the brain, there is this particular two set of nerves, they cross each other, and that is known as the optic chiasma. It is located just below that, and it is connected to the infundibulum by the means of stalk. This may be absent in some species. In mammals, you have seen that you have an infundibulum and a stalk. This may be absent in some species. So just to show what is the infundibulum, I will try to find out a proper image. Pituitary structure. A normal pituitary gland, not a fish gland, but a normal pituitary gland image. If you have to look at a normal pituitary image. So you have the hypothalamus, from where neurosecretory cells enter into the neurohypophysis. And you have the adeno-anterior lobe of the adenohypophysis. This is not a better picture. I'm looking for a different type of picture. Let's see what type of picture it is. This is also not a good picture. So this is the structure. You have the pituitary gland over here, and this is the neurohypophysis, this green part here, and this side is the adenohypophysis, and this is coming from the hypothalamus through this particular stalk. This is the infundibulum, and it enters directly into the pituitary. So let us get back to our studies. So you have the, let's talk about the rostral pars distalis, this region, the rostral pars distalis. This region we are talking about, the rostral pars distalis. So it is called also the anterior most part of the adenohypophysis, and it has got three types of cells. Most of the cells are acidophils. They are stained by azocarmine or orangey. They may show follicular arrangement surrounding the blood capillaries. So you can see that this is like a thyroid follicular arrangement surrounding a blood vessel which is in the center. There are a few cells which are stained with aniline blue and with aldehyde fuchsin. PD sir will explain all these things. And they are known as cyanophils. A few non-granular cells that do not stain also are present, and they are known as chromophobes. They don't take any color. Then you have the proximal pars distalis. The proximal pars distalis. Here. This is the pars intermedia, this region which I have just currently drawn. I'll just use another color and explain. This is the proximal pars distalis. And it lies between the rostral pars distalis and the pars intermedia. But it is not clearly demarcated from the rostral pars distalis. But it is different from the pars intermedia. You can see it's because there are large number of large ramification of pars nervosa. So lots of nerve endings enter into the pars intermedia. So this proximal pars distalis, they contain acidophil cells, cyanophil cells, and chromophobes, and their size may vary during different times of the year. During maturation and spawning period, the cyanophils are more numerous. So during the sexual development and when they are about to release spawns, the number of cyanophils are much higher. But during rest of the year, acidophils are large in number. So these acidophils can be stained with azocarmine. You have to remember which stains, or orangey stains. The cyanophils are stained with aniline blue, PAS, and aldehyde fuchsin stains. Now we go to the next part, which is the pars intermedia. It is the posterior part of the hypophysis, and it is extensively supplied by branches of pars nervosa, branches of nerves. The branches of nerves. So they are basically cyanophils that are stained with aniline blue, and acidophils that are stained with azocarmine or orangey. Some species contain amphiphil stains that take both the blue and orange stains. Both the two stains have been taken. Next, we come to the neurohypophysis. So there are a large number of nerve fibers which arise from cells in the hypothalamus and enter the hypophysis. So let's have a look at the picture again. Lot of cells, the hypothalamus may be somewhere over here. From the nerves, lots of nerves enter over here. These are nerves endings are coming into this neurohypophyseal region. This is the neurohypophyseal region. The large number of nerve fibers, they enter here. These fibers then branch repeatedly and they are connected with the pars intermedia. So there are connections from the neurohypophysis to the pars intermedia. There are cells, the pituitary and droplets of neurosecretory materials are seen in these fibers. Lymphocytes and migrating cyanophils have also been reported in some species. They can be stained with aldehyde fuchsin, chrome alum hematoxylin, and aldehyde thionin. Some herring bodies are also found. These are largely stained. They're neurosecretory materials. So these herring bodies actually gave the concept of neurosecretion. Now you know that what are the hormones of the pituitary gland. So at least there are 9 hormones comparable with those of mammals are produced. Prolactin, somatotropin, which is the growth hormone, gonadotropin, TSH, adrenocorticotropin, intermedin, vasopressin, and oxytocin. So these hormones are present. So let us talk what the gonadotropes do. We have seen the where the gonadotropes are located. They are situated in the proximal pars distalis. They contain glycoproteins and they are stained with pars, alcian blue, aniline blue, and aldehyde fuchsin. Two types of hormones have been suggested. FSH and LH are secreted in teleost, like mammals. This is the gonadotropes. Multiple gonadotropins are secreted in the teleost. They are divided into two groups: hormones rich in carbohydrates, which may be involved in steroidogenesis, permeation, maturation of ova, and ovulation. And the second group is poor in carbohydrates and they are involved in vitalogenesis, that is the production of yolk. Then you have the thyrotropes or TSH cells. These cells are found in the proximal pars distalis and they are basophils and they can be easily distinguished from the gonadotropes just on the staining affinity. So you know what the TSH does. It actually goes and stimulates the thyroid gland to secrete thyroxine. Stimulated to secrete thyroxine and thyroid hormones. Then you have the somatotrop cells. Pars distalis that is found. Two types of acidophils can be distinguished. Cells take orangey stains and are somatotropes and found in the proximal part distalis, which is intermingled with thyrotropes. The second type of acidophils, they take azocarmine or erythrocin and they are the prolactin cells. They are the prolactin cells. So, as you know, they are responsible for the growth. It is the growth hormone. Then corticotropes or ACTH cells, they target the adrenal glands. They are the target organs. Then you have the prolactin cells, lactotrophs, they are sometimes known as. These cells are located in the rostral pars distalis. And these are the erythrosin, azocarmine, acid fuchsin, but they're negative to PAS. Now let us look at the hormones of the pars intermedia and the neurohypophysis. Two types of cells are present in the pars intermediate. There are two hormones, MSH and MCH are released. These are the melanophore dispersing hormone and the melanophore concentrating hormones, which influence the pigment of the skin. Skin becomes darker or lighter based on effect of these hormones. So MSH, concentrating hormone, the MCH, what they will do, they will concentrate it and the skin will become darker, or that region will become darker, and dispersing hormone, it will make it lighter. In the neurohypophysis, you know two hormones, arginine vasopressin and isotocin. Vasopressin, oxytocin, they are all in this region. It constricts blood vessels and enhances water retention by kidneys. So let us look at the neuroendocrine, briefly the neuroendocrine regulation of pituitary gland in fishes. It's the same as that of mammals. Studies have shown that two regions, the nucleus preopticus NPO and the nucleus lateralis tuberculis NLT, they are the important nuclear areas which control the activities of the pituitary. NPO is dorsal to the optic chiasma and its axons extend into the pituitary gland. It stimulates the pituitary gland to secrete. And you have the NLT, it lies along the ventral surface of the infundibulum and its neurons innervate the cells of the hypophysis. As we know that the hypothalamus contains with the help of neuroendocrine hormones through the hypothalamo-hypophyseal blood portal system. So you have releasing hormones through the hypothalamo-hypophyseal proteins. They secrete pituitary hormones. And these pituitary hormones go to organs, target organs, and hormone is released. Other hormones like thyroid, thyroxine, adrenaline, all these are released. Pituitary gland appears to be regulated by this gonadotropin releasing hormone, RH. It is identified in different locations of the teleost brain. Besides, a gonadotropic releasing inhibiting hormone, GIH, it is also present in the brain of teleost. Even you have the thyroid stimulating hormone, TSH, ACTH, all the cells, the same mechanism. You have a releasing hormone from the hypothalamus, it adenohypophysis stimulated. You have ACTH, you have TSH, etc. All these hormones are released from the adenohypophysis and then it goes to the target organs, which may be adrenal, thyroid in this case. You have the corticotropin releasing factor. You have different releasing factors. You can even study your semester 2 physiology note, endocrinology note. You can find it over there. All the same hormones are present. So there is a prolactin releasing inhibiting hormone, PIH. This is also have been reported. Prolactin releasing hormones have been reported. And interestingly, the extracts of the pituitary glands, they are being used in induced breeding of fishes in fish culture. We have this chapter, induced breeding. So this is about pituitary gland. Let us go to the thyroid gland. So let me. I've saved a picture to show you where the thyroid gland is located in the fish. So this is the ventral aorta. What is labeled over here is this is the ventral aorta. Along the ventral aorta, these little circular things you see there. This is the brain. Brain is over here. This is the fish. You see these little dots. These are the thyroid follicles in fishes. Just to understand, I will send this picture also, just to understand where it is located. They are present in a diffused condition. They are present in a diffused condition around the ventral aorta. We have seen it in the diffused condition, that is dot 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 along the ventral aorta and the afferent branchial arteries. In few fishes like parrot, swordfish, the thyroid, it is a compact and encapsulated gland that is also found. So the gland may be diffused or encapsulated depending upon the species. Depending on the species. The air breathing fishes যাদের অতিরিক্ত শ্বাসনালী রয়েছে like Heterocystis fossilis and Chana striatus or Batrachus, Clarius batrachus, they possess a compact and encapsulated thyroid near the anterior part of the ventral aorta. So it is not like these dot 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 dot. They have actually a gland, not dot dot follicles. So as you know, each follicle, it is a hollow ball consisting of a single layer of epithelial cells surrounding a fluid-filled cells. Remember the thyroid follicle pictures. So this is the ventral aorta. Along that you have these thyroid follicles present. You have first you have a single layer of this is single layer of inside this region you find the colloid present. Same as with the mammals, you can draw this. This is the single layer of epithelial cell. Let's read the thyroid gland in your second semester BSc physiology. Single layer of epithelial cell. Right. So this is how the follicle and the colloid is released inside. It is stored over here, and gradually the colloids, triiodothyronine, and all these things. And the function, it is to the hormone contains monoiodothyroxine, MIT, DIT. You see that these two. This is MIT. You have iodines attached to here. Two of them join and form DIT, and that is actually nothing but T4. So if this is DIT, if this is a thyroglobulin molecule, one iodine on this is MIT, and another if you have two iodine attached, this is DIT. So and then if something like this, it becomes this plus MIT plus DIT, you can have T3. Two DITs together, they will form the T4 or thyroxine. So all these hormones are present, and what is the function? same as the function of the thyroid. Important role in oxygen consumption. Then thyroid influence the metabolism, carbohydrate metabolism of the fish. Then growth of the fish. It influences osmoregulation. It has important role in reproduction and migration of the fish. In migration of fishes, you will find this particular thing. So these are the function of the thyroid glands. Essentially, this actually finishes this endocrine part. Do you people have any questions? Please feel free to ask. কোনো প্রশ্ন আছে? আছে প্রশ্ন? Any questions? Hello, am I audible? Hello.
Yes, sir.
Okay.
Yes,
No
sir.
questions then. Okay, there are no questions. Okay, then I will again next year we will talk about the lateral line system. That is a very interesting part. I will send you the note. Study those parts. You have already got the notes on the digestive system, and now I will send you the note on the lateral line system, electric organs. All these things are there in my portions. Okay, bye bye then.